Okay, so this is the first in a series of tutorials on SpliceSeq. SpliceSeq is a freely available tool for the evaluation of alternative splicing in RNA-seq data. Changes in splicing patterns are fairly common, particularly if you're looking at different developmental stages or different tissue types or disease progression. And SpliceSeq is a nice tool for letting researchers explore and identify splicing changes. SpliceSeq was developed by In Silico Solutions in collaboration with the Bioinformatics and Computational Biology Group of MD Anderson Cancer Center with funding from the Chapman Foundation and NCI's TCGA project. So in this tutorial, I'm going to show you how to run SpliceSeq and also take a look at some RNA-seq data with the tool. First, one slide of background information. The central structure for analysis in SpliceSeq is a splice graph. So for every gene, we assemble a splice graph from its known transcripts. In this example here, there are 4 transcripts for a gene. You notice there's 2 different transcript starts for the gene, 2 different exons where transcription starts. We have an alternative splice event in the middle where sometimes this exon is included, sometimes it's excluded. And finally, we have an alternate donor site here in this exon where sometimes the near splice signal is used and sometimes that one's bypassed and a later splice signal is used. If we assemble that into a splice graph, it looks like this. So, our 2 different start sites, exons 1 and exons 2, are spliced into the central portion of the transcript. Then we have this representation for our alternative exon. Sometimes exon 4 is excluded, sometimes it's included. And finally, the alternate donor site is represented like this with a single long exon split into 2 sub-exons, 5.1 and 5.2, with splices coming out of each one. So this is what our splice graph looks like. The splice graph structure has a couple of advantages in looking at alternative splicing. The first is that it does a really nice job in the alignment phase of handling little exons with complex splicing patterns coming into and out of it. Also, splice graphs for the longer transcripts deal very nicely with the ambiguity that we get with alternate splice events, different splice events on the transcript that are widely separated. And this splice graph gives us a very concrete way of representing the data as it was actually observed rather than getting into a bunch of inferences on that. And finally, the splice graph serves as a common point of reference that we can use for comparing samples and grouping samples and comparing groups of samples. So splice graphs are the structure that we use, and I think they do a real nice job. So let's take a look at the tool. So the easiest way to run SpliceSeq was generally just to enter SpliceSeq into search engine, and the first result in Google anyway is the MD Anderson SpliceSeq site. And this site has lots of help information on the various views in the tool, the methods, how to install it, how to load your own data. What we're going to do for today is to just click this launch button right here, which is going to run SpliceSeq as a Java Web Start application. To click a couple accepts and runs, and then SpliceSeq will come up. So this is the SpliceSeq tool. By default, when you first run it, it's going to come up pointing to a set of data that we have loaded on our servers from a publicly accessible study on different tissue types. So the first thing we want to look at in the tool, or make sure you're aware of in the tool, is which view you're working with. There's a bunch of different views here. For this tutorial, we're going to work on just the sample view, which lets us look at an individual sample at a time. So we're going to go ahead and click Sample View. There's a progress bar down here. When there's a lot of data coming down, it'll take a little while for the data to come down, but you'll see the chunks of it coming down, and you can work with it while it's coming down. We're going to switch to the Gene Summary tab. There's a couple other tabs here, but we're going to work on Gene Summary today, and I'll cover the other ones in a different tutorial. The samples that are loaded in various studies, and within each study you can then see the individual samples for that study. So we had 2 studies there, and we're currently looking at this heart tissue. So in the heart tissue sample, if we want, we can go to favorite gene, hit enter or hit this to jump to the gene, And you can click down here on the graph to zoom in and out. If you zoom out, you can sort of get a whole feel for it, but it's a little harder to see. The coloring or shading is done based on how many reads we saw for the sample. So the darker ones had more reads, and the lighter ones had less reads. If it's a dotted line, there were actually no reads in the sample that we're currently looking at. The exons are drawn to scale based on how large they are, and the portions that are drawn thin are non-coding. The thick parts are actually the coding region. So here we have a start of transcription, and then the protein coding starts here and continues on down as we splice through to this last exon, stops, and then we have a little more UTR region here. We can toggle this to actually take a look at the read values. So that was in this control panel here on the left. And now we're actually looking at the read counts, the raw read counts that we saw. So we had 364 reads that were on exon 2. We had 16 reads that crossed the junction between 2 and 3, and so on. So raw reads are kind of nice to see, just to get down to the very bottom of it. But generally you would want to work at the corrected ones, the normalized ones. So these are normalized based on the length of the exon and then per million reads in the sample. And so if we look at this, we saw 364 because this is such a long exon. We had 95 on the neighboring shorter exon. Once we've corrected that for the length, we get values that are at least kind of in the same ballpark and allow us to compare across exons. Okay, so and here we see this promoter, exon 2, being used in heart tissue much stronger than the promoter 1. We had almost no reads on this promoter, or this first exon. Look at one other gene here. And in this exon, the interesting part is down here, or this transcript, this gene, the interesting part is down here in exon 6, where in heart tissue, it prefers to exclude exon 6. Almost all the reads go around exon 6, and we had maybe just a read or two on exon 6. So now let's switch over to brain tissue. We're going to take a look at that exact same gene. And we can see that in brain tissue, this pattern actually reverses. So there are still some transcripts that are excluding this exon, but the vast majority are actually including exon 6. So nice example of tissue differentiation here. And we'll look at that other one that we looked at. Oh, here on the right-hand side, you get a breakdown of what the gene does from gene cards. So we have the uniprot and the NCBI description of the genes. So that's kind of nice when you're jumping around and you hit an interesting event, but you're not quite sure what the gene does. If there's something really interesting that you do find and you're curious about how the splice graph here in SpliceSeq relates back to other gene models, you can come over to this exon info tab, and that'll provide you with the genomic coordinates of each exon, and you can even cut and paste out the sequence of a given exon. Right click and then left click to copy it out, and that'll let you design primers if you wanted to do some PCR. So last thing we'll do is look at that one. Here's another difference. We've switched over to brain. So in brain tissue, we have a reversal on the first exons as well. So it's in brain tissue, we're getting a different promoter on that first gene we looked at. It's using exon 1 as its first exon as opposed to exon 2. Okay, so that's just some quick examples, and I'll give you a little bit of feel for how you can move around within single samples, take a look at your genes of interest, and understand the splice graphs. We'll have a couple more tutorials on doing comparative analysis and grouping samples, and also how to load your own data and how to look at the protein impacts of splicing changes. So that's it for the first tutorial. Thanks.